Okay, hello everyone. Today we are going to discuss important topic, okay, and related to my previous class in future star coaching classes. If you are subscriber of this or biodigen, so in my previous class I have explained the flower. So in flower we have seen that there are male part that is called endoecium and one of the male part is called stamen. Okay, so this is stamen. A stamen is further, it is made up of filament, a long stalk like a structure that is called filament, which is connected with the help of connective with two lobe structure which is further diathecous. So altogether there are four chambers you will observe in the anther or pollen chamber. Four chambers are being seen. So in picture you are already observing. So when we take a cellular diagram from a portion of the pollen chamber, we will see that there is an outer layer in cellular diagram. The outer layer dumbbell shaped structure. These are the cells which are dumbbell shaped and known as epidermis. Always epidermis is dumbbell shaped. So outer layer is epidermis. Below epidermis there is a endothecium. Below endothecium, third layer is middle. So first is epidermis, second is endothecium, third is middle layer, and the fourth is tapetum. From outside to inner side in this chamber I am talking about. From this side to this side I am going from inward. So outside to inward. And tapetum serve as a nutritive function. Then below the tapetum there is a sporogenous tissue. All are diploid since they are grown from seed which is diploid form after fertilization. So all are diploid having 2n chromosome diploid. Now this sporogenous tissue known as pollen mother cell, also known as pollen mother cell, this tissue is also known as pollen mother cell. This undergoes reduction division. These sporogenous tissue diploid myocytes undergoes reduction division to form 4 haploid pollen tetrad. 3 are observing the tetrahedral structure and 1 is hidden. So not it is 3 cell, it is 4 cell, 3 are seen and 1 is hidden. In the 3-dimensional picture, I can't show the 4th one. But these are the 4th. You can understand this like this one. Okay. 4 different type of pollen grains, future pollen grains, pollen tetrad. They are called tetrad. Tetrad means 4. But 3 are you observing, 1 is hidden. Now this each pollen tetrad, out of pollen tetrad, each one developed into pollen grains. Okay, that modified changes into a matured pollen grain. So, if the question comes that label the cellular diagram, this let me tell you for CBSE, if you are the student of again CBSE in this year, CBSE 226, then this question is very VVI, very important. We guess that this question will come. So try to understand properly. So up to this, I think no problem is there. Now, how can you define microsporogenesis? Microsporangium is the pollen grain. Okay. So enter the process, here the process of formation of pollen grains by the reduction division in the pollen mother cell, which are myocytes, is known as microsporogenesis. The process is called microsporogenesis. What happens? The stamen, inside the stamen, as we have seen, the pollen chambers are there. In pollen chamber, the diploid cell, myocytes, pollen mother cells are there, which are also known as a sporogenous tissue, a spore giving tissue, undergoes reduction division to form pollen tetrad. And this pollen tetrad out of 4, but each one each of the tetrads developed into mature pollen grains by changing its form, which got at the maturity exine, intine, and this one is the vegetative nucleus, and the smaller is the, which is floating on vegetative nucleus, is the generative nucleus. So this one generative nucleus, vegetative nucleus, or cell, you can say vegetative cell, generative cell, and the nucleus is vegetative and generative nucleus. Now, this generative nucleus migrate immediately after pollination. At this stage, the pollen grains, two-celled structure, the pollen grains is pollinated from anther to stigma, by the cross-pollination or self-pollination. And then the stigma, right pollen grains are nurtured, are supported by a stigma of their competent flower, and after getting nourishment, the pollen tube developed where the exine, which is a very resistive layer made up of sporopollenin, which is very resistive, the most resistive substance in the biological world, most resistive, NEET का क्वेश्चन है ये, याद रखिएगा, most resistive substance, the sporopollenin, resistance to what? Acid, therm, heat, base, dryness, all resist them and protect them. They are spinous. So where it is lacking, the pollen tube, that is called germ pore. Where the sporopollenin is lacking, the area is called germ pore. So through germ pore, the vegetative cell pushes the generative cell, or the nucleus of generative cell migrate here. After migration of nucleus, it divides into two male gamete mitotically. Remember, meiosis taking place once in the life, second time meiosis cannot take place. So after dividing mitotically, two male gametes are produced in the pollen grain, which are meant for double fertilization. So this is all about your microsporogenesis. Easily you can write and explain. Okay? Any question if you have, you can inquire the question at our the coaching center. Future, I am also associated with Future Star coaching classes. My channel is Bioaddition. So in Bioaddition, maximum video I have already explained. You can go and download there and see. No need to go elsewhere. Such type of topics will always be discussed. But remember, in Future Star coaching class, I only give the time for Thursday. Thursday याद रखिएगा. थर्सडे को मैं शाम में देता हूँ यहाँ 6 से 8 बजे कभी मिल सकता है. और संडे को आपको मैं देता हूँ यहाँ टाइम, तो संडे को आपको मिलेगा 10 से लेकर के 1 बजे तक 1 PM तक मैं रहता हूँ. तो इसमें कभी भी आपको क्लास मिल सकता है. अकॉर्डिंग टू नीड हम लोग क्लास इसमें देते हैं. ठीक है, आप रेडी रहिएगा, सब्सक्राइब कर लीजिएगा और पढ़ते जाइए, कोई दिक्कत नहीं है. थैंक यू. कोई प्रॉब्लम होगा तो पूछना. देखिए सर.